In record time, scientists have gone from harvesting antibodies against SARS-CoV-2 from survivors of coronavirus infections to testing the antibodies' safety as a drug in humans. Now, before we start, make sure you hit the subscribe button, and if you already did, do click on the bell button so that you will not miss out any of our new videos. Thanks to a remarkable feat of natural molecular engineering that allows our bodies to generate countless different kinds of antibodies against pathogens, COVID-19 survivors typically have scads of SARS-CoV-2 specific antibodies cruising through their bloodstreams. Only a few of those antibodies stick to the virus at the right spot, blocking a protein the virus needs to break into cells, while other antibodies bind to it without stopping infection. By the time a human body has generated these diverse antibodies in sufficient quantities, the original infection is usually nearly over, but the antibodies remain, leaving the immune system braced for a second infection. For the past few months, scientists have been eager to find the most effective antibodies the human body produces and turn them into drugs. In contrast to convalescent plasma therapies, whereby a hodgepodge of antibodies from recovered patients is given to people battling COVID-19, such monoclonal antibody treatments would be standardized, manufactured at scale, and potentially more effective. The treatments could be given to people in the early stages of COVID-19, or used prophylactically to give instant, short-term immunity to vulnerable people such as healthcare workers or those in which vaccines may be ineffective. Technological advances have allowed scientists to find promising antibodies at a record pace. Just this week, June 1st, the pharma giant Eli Lilly announced the launch of the world's first human safety trial with a monoclonal antibody against SARS-CoV-2. According to Dennis Burton, chair of immunology and microbiology at the Scripps Research Institute in California, before there's a vaccine, there could be great use for the antibodies in prevention. And even when there is a vaccine, they could be used in therapy. But first, all of these things need to be tested in humans. Choosing the best fighters, Vanderbilt University immunologist James Crow was already working on a long-term project to develop monoclonal antibodies against potential epidemic-causing viruses in late January when it became clear that SARS-CoV-2 was spreading into the US. His team rushed to track down people who had recovered from the infection, ones who had been infected in China who then traveled to the US, and procured blood samples. Using several commercial technologies, Crow and colleagues screened millions of white blood cells to pick out plasma B cells that carry SARS-CoV-2 specific antibodies by teasing out the cells with viral antigen. His team then extracted antibody-encoding DNA from those cells and used that as a blueprint to synthesize them into antibodies. Through experiments testing whether the antibodies could prevent SARS-CoV-2 from entering a human cell in vitro, they identified around 400 kinds of antibody that recognized the virus's spike protein, 50 of which could neutralize the virus in vitro. Several companies Crow has partnered with, including AstraZeneca, the Nashville-based biotech company ID Biologics, and Ology, a company contracted by the US government to develop and manufacture monoclonal antibodies for COVID-19, are planning clinical trials for some of the candidates in July or August. They literally handed them off to manufacturers within the first few months of an outbreak. That's what's different now, is that these technologies have matured to the point where they can compress the time down to weeks, whereby this type of thing used to take years. Carl Hansen, CEO of the Vancouver-based biotech company AbCellera, which has partnered with Eli Lilly to develop its candidate, explained that the antibody that Eli Lilly is currently administering to COVID-19 patients, LICOV 555, was also discovered in some of the first COVID-19 patients in North America. Hansen said it would normally take anywhere from 2.5 years to 5 years for a program like this to move forward. But thanks to a rapid antibody identification platform AbCellera, whereby scientists have refined as part of the pandemic preparedness program funded by the Defense Advanced Research Projects Agency over the past several years, the researchers were able to go from a blood sample to a clinical trial in just 3 months. Others are pursuing different strategies. One group of scientists at San Francisco-based Biotechnology turned to antibodies isolated years ago from the blood of people who had recovered from the original SARS coronavirus. They recently found one such antibody that also neutralized SARS-CoV-2 in vitro. According to the company's CEO, Jake Glanville, who is working with several academic labs and a Department of Defense laboratory to test his top candidate in hamsters, this San Francisco-based biotech startup Distributed Bio used computational methods to tweak anti-SARS-CoV antibodies, so they're adapted to the novel coronavirus, among other methods. The German biotech company UMAB used a library of antibody protein encoding genes generated from hundreds of healthy people and shuffled the genes in different combinations to create a diverse range of antibodies, essentially mimicking the human body's own strategy. The team then used a technique known as phage display to screen which antibodies would bind most strongly to SARS-CoV-2 protein. According to Andrei Frenzel, the chief scientific officer at UMAB, combined with studies of COVID-19 patients' blood, the group found several antibodies that looked promising, and are currently testing their efficacy in vitro. Despite being in different stages of development and using different methods to find them, these groups' antibody candidates are similar. The ones that are most effective in neutralizing SARS-CoV-2 block the receptor binding domain, RBD, a dongle-shaped protein at the end of the virus's spikes that it uses to dock onto the ACE2 receptor of human cells. Eli Lilly's antibody candidate also binds to the spike protein. Erica Ullman-Sapphire, an immunologist at the La Jolla Institute for Immunology, said that their prior studies have suggested that blocking receptor binding is a surefire way to neutralize the virus and come up with a protective antibody. Sapphire recently launched a consortium of 50 different startups, corporations, and academic labs that have agreed to send their best antibodies to her lab. Her team will then line up the antibodies side by side and identify the very best ones. The goal is to find antibodies that neutralize the virus at the lowest concentrations, which will help reduce the typically sky-high costs for such treatments and make them financially accessible for low- and middle-income countries. Aiding her team's investigations is an 11-foot-tall cryogenic electron microscope, which can visualize, at the resolution of a couple of angstroms, the precise molecular interactions between antibodies and SARS-CoV-2. Such studies will help scientists identify what exactly makes winning antibodies effective, examining, for instance, the angles from which they approach the viral surface, and which antibodies enhance each other's efficacy. Sapphire said one can actually look at what a successful antibody is and how it works. Antibodies for prevention or treatment. In Eli Lilly's Phase 1 trial, 32 COVID-19 patients in the US will receive varying doses of the antibody candidate, Lykov 555. The study only aims to assess side effects and tolerability, and results are expected by the end of June, according to the press release. This and upcoming trials may also shed light on the possibility of a rare and poorly understood phenomenon known as antibody-dependent enhancement, in which antibodies can worsen viral infections, although there's no indication this happens with COVID-19 and prior coronavirus exposures or experimental vaccines. Should the study go well, the company plans to proceed to a Phase 2 trial to test whether the antibody is effective as a treatment, and also to test whether the drug may be promising in preventing SARS-CoV-2 infections in vulnerable groups. According to Burton, who has experimented with one of his own candidate antibodies in hamsters, some groups have tested that concept for SARS-CoV-2 in animal studies. He further stated that untreated rodents lose around 10 to 15% of their body weight when infected with SARS-CoV-2, but if they gave them a neutralizing antibody before infecting them, they were fine, they didn't lose any weight. In people, they would hope that they would stop the virus causing serious disease at least. Using 3 different mouse models, Crow's collaborators have also found that some of his antibody candidates appear to work for prevention or treatments. While some companies are focusing on administering individual antibodies, others, such as the New York-based biotech Regeneron, are pursuing cocktail treatments. Hansen said that a single antibody may be easier to manufacture, but a combination could be important should SARS-CoV-2 mutate in a way that makes one antibody ineffective, although there's no evidence of that yet. Eli Lilly aims to test both single and multiple antibody therapies. Sapphire said opting for a cocktail strategy would also be wise if it turns out that SARS-CoV-2 is capable of using multiple ports of entry into a cell. Viruses are crafty and many of them have more than one way into the cell. Researchers can't yet rule out that possibility for SARS-CoV-2. The big challenge: making antibodies at scale, affordably. In the past, scientists made antibodies by fusing B cells with a cancer cell line to make them immortal so the cells will produce antibodies indefinitely. But modern, more efficient methods have replaced that strategy. Some of Crow's partners are using tobacco plants that are genetically modified to produce SARS-CoV-2 antibodies. By far the most popular method is to make antibodies using a cell line derived from Chinese hamster ovary (CHO) cells, which can be grown in giant bioreactors, and the antibodies they produce can be sifted from the culture. However, that manufacturing process is relatively slow. Frenzel said it can take many months to set up a productive CHO cell culture and scale it up in large bioreactors, although several groups are exploring ways to speed up that process. Hansen stated that the ability to scale up treatments will depend on just how much will we need and when will we need it. If the antibodies will only be used for treatment, it's unlikely there will be shortages, but if there's no vaccine soon, and policymakers decide to give antibody therapies as preventive therapies for large numbers of the public, manufacturing capacity may reach its limits. According to Eli Lilly, the company has begun manufacturing its antibody in bulk and aims to have several 100,000 doses ready for COVID-19 patients by the end of the year. In addition, the process of growing these cell cultures, as well as the research to identify and develop the most effective antibodies, can make such therapies extremely expensive. According to a 2018 study, the average annual price of antibody therapies for cancer and autoimmune diseases between 1997 and 2016 was nearly $97,000. If costs for the first coronavirus antibody treatments push into the thousands or tens of thousands of dollars, it raises the question of who will pay for them. Frenzel speculated that perhaps some governments may be willing to cover, or at least subsidize, the costs of such therapy, favoring it over suffering through the economic disruption of another lockdown. During a global pandemic, the treatment has to be accessible for developing countries, too. Sapphire said it is abundantly clear now that infectious disease anywhere in the world can become infectious disease everywhere in the world. She further said that we are not safe until everyone has access to protection, and so one way or another, we're going to have to get there. One problem with antibody therapies developed against Ebola virus is that although they were effective enough to save patients if administered in time, Glanville of Distributed Bio stated that they were way too expensive to be able to treat a disease which was happening in the developing world. Eli Lilly did not provide details on the possible cost of such treatments, but Nicole Hebert, a spokesperson for the company, stressed that the priority is finding a treatment for COVID-19, rather than making a profit. Hebert wrote to the scientist in an email stating that, regarding access, the industry is working with governments and insurers to ensure that when new treatments and vaccines are approved, they will be available and affordable for patients. He aims to charge no more than a few $100s a dose, which should suffice as a treatment and provide protection for around 2 months. Glanville further stated that he don't know the exact number, but he is not going to be charging $8,000 a dose. He is going to keep the number deliberately accessible so the whole world can gain access.